Okay, let's get started. Yeah, I guess we're now always here in this room. From now on, no longer upstairs, because it's now reserved permanently. Yeah, homework, has anybody tried it? Okay, have you got through with it easily, or what were the difficulties? Let me quickly open our homework again. I mean, it was basically just going through the different steps of the analysis and trying around a bit. And I thought maybe we just do it together, and then we can check which questions appear in the process. Now, as you've seen, I've already did the homework here because you see all the results, which is unusual. Usually I post homeworks, never do it myself, and somebody else does it. And but so I could show you the plots already ahead. So they are all in here, it's only that the code is hidden. Let's see if I find it. Render. Okay, files, homeworks 2. Where's homework 2? Homework pet, I think I called it. Yep. Okay. So sorry, I'm feeling a bit slow today, but we'll see what's happening. Good. Okay, for those of you who are new to R, maybe that's something worth pointing out. R has this notebook style, which is very useful if you want to make documents where you mix code and analysis. And you've seen this is how I made all these things, where you always see these code blocks, text blocks, and then below wherever the answers of R. And the way how you do this in RStudio is by making this what they call quarter documents, and where you see how these quarter documents work. I type all this text, and then occasionally I can type triple back quote, and then in curly braces R, and again triple back quote, and then it will put a code chunk in here. And you can use this markdown notation, where, for example, this means a level 3 headline, or this here means make a hyperlink. Everything in the square bracket should be what's displayed, and the thing in the round parentheses should be what's labeled. And as you can see, this is what we then got out of that in the homework. But here, this is this level 3 thing, and here you can see the hyperlink, or here you can see something like this, which I put in backticks. So that's quite useful to quickly make a nice little document that you can then turn into an HTML or a PDF file, or also into a LaTeX file, I think, and compile it. And so here, this is the first thing, and as you see, usually it looks like this. But I had added this echo is false so that it doesn't display the code, because this means that you don't see the code here and have to figure it out yourself. But of course, it's nothing deep here. We simply call the read matrix market package to call in this count matrix thing. Now we can already make a short stop and have a look at this count matrix and inspect it a bit, because it's always a good idea when you read data to first have a look. I simply say CM, and it shows me here what I have. And as you can see, we see here our big matrix. It has this class DGT matrix, which means it's a sparse matrix in triplet or T form. That means internally, if we look a bit at how this CM is stored internally, it has here these 3 vectors. There's a vector dim, which tells us the dimension. I could also simply say dim CM, and it tells me that our matrix has 36,000 rows for the 36,000 genes and 1,100 columns for corresponding cells. What we can also see is now that it has 3 big vectors, all of length, what is this, 24.3 million. And these vectors are labeled I, J, and X. And this is very simple. It simply means the matrix is all zeros except for a 1 at row 24, column 0, another 1 at row 48, column 0, and 9 at column 170, row 170, column 0, and so on. So it's simply a list of coordinates which tell us where the non-zero matrix is. And obviously for a matrix which is 99% zeros, this takes storing the coordinates plus the values takes less space when storing everything, because this way it's times 3. This thing is, I can't, 1e6, this thing is 72 million numbers, and if we were to calculate, to store the individual numbers, then you would get, of course, 1e6, 420 million numbers. So we save space, which is important, not only to save space on your hard disk, but more important to save space in your RAM, in your memory. And the reason why this is important is, well, these things can get big quickly. So if we have a look here of what R tells us, we've already used so far 1 gigabyte of memory. My computer has a total of 16, so you can quickly run out of that, especially if you have an older computer with only 8. You had a question? The other thing is, for calculations, such matrices might be easier to use, because imagine you want to multiply, you want to make a matrix vector multiplication. You have a vector which you want to multiply from the right against this matrix. When you have to do your usual every row against every column thing that you do in matrix or matrix vector multiplication. So the computer has to internally go through the matrix and vector, say this times that, and so on. You know how you do it. When also you do it by hand. And most of the time the computer calculates 0 by 0. It looks up and sees a 0 in the matrix and says, okay, I add 0. And this, of course, takes way longer than if a computer only goes through the non-zero values. If you want to make a matrix vector multiplication algorithm with this kind of matrix, then you would simply go through all these values, always wonder about, okay, this is in row X, in the row I, so it contributes to row I of a result vector, and we have to multiply it in column J, and you can easily see how you can make a much faster multiplication operation. So using sparse matrices not only saves memory, but it also saves time. But it saves the time only if you change all your standard linear algebra algorithms by those which can deal with this kind of information. A technical thing, which is of course, usually I would skip over it, but we are a technical course here. As you might notice that all of these, that the column names and row numbers here start with 0. But R is a 1-based language. In R, all row and column indexes start with 1, while in other languages like C or Python, they start with 0. This is because internally R uses here a general sparse matrix library, which originally was written for C, and R simply wraps around it, and this is why internally it needs to it wraps all this. So if I now want to see, for example, this 9 here, and I say 170,0, then as you can see, now something strange has just happened, because it said that it has done a sparse dense coercion and now it tries to put everything into this big thing, and my computer blows up, but I don't know why. It's a good start, but my computer crashes immediately. Let's restart R quickly. If you work with this big data, you will notice R can sometimes get quite annoying there, so we have to be a bit patient, but we have time now, I hope. How did it work for you? Did it work smoothly, or did your computer also crash all the time? Crashed a lot? Yes. This is what you can now already see. It's currently restarting R and it's loading the memory. So what I will now do is here, I say pkill R session, and this kills my R session. So I now get a new one. And yes, you can already see my idea is to use these exercises to also look a bit at practicalities of how you work with this stuff, because that's something you usually need a lot, and that typically often takes more time than actually understanding the computer science side of things, or the mathematics side. So, here we go. I try again loading my count matrix. Sorry that things are a bit slow, but let's try again. strcm, here's the count matrix, and now I say something like 171, oh, this is it. If I try to type, it tries to help me by immediately looking something up. This is not good. Anyway, what I wanted to say, if I now were to look here at element 170,0, I would get an error, because that matrix is, because it doesn't even know what this is, 170,0. But if I ask for 171, because this is what we actually have. But now I won't try to look into this anymore. Because the issue is, what has just happened is RStudio tried to be helpful and tried to help me by already looking into the matrix and checking out to what I might want to type, always typing ahead of a user thing. And for this thing, it needed to look into the code which looked into the matrix was written for normal matrices, but it found a sparse matrix, so it decided, let's quickly switch it to dense so I can look and help the user. And switching it to dense, of course, blew up my memory. The RStudio people are currently very busy trying a lot of these new features, and I think the last update of 2 months ago was a bit introduced a lot of stuff which doesn't work properly. So if at the moment you're new to RStudio and you get annoyed, this seems to be an effect of the last couple of months. It will hopefully be better again soon once we fixed that. So, but here we have the count matrix. We can look this way. For example, I like to always have a look in the top left corner to get a quick overview, even for I only see a single one. And the next thing I now want to do is I want to label the rows and columns, because so far we don't. You remember we had in this we had these 3 files in here. Let me look again at the 3 files. I go here to tim/filtered matrix, and we just read in the matrix file, which here has all these values: 25, 1, 1, 49, 1, 1. You can here, by the way, already see the matrix format seems to be 1 based. When it read it in and changed it into 0 based, so that the C code can work with it, and when displaying it to us R users, it adds it to 1 again. So there is a constant moving back and forth of adding 1 and subtracting 1, which can drive you quite crazy when programming with these things, because you never quite know whether things are 1 based or 0 based. Anyway, so this is how these things work. But we have also these other 2 files, namely the file barcodes, which are the labels for the columns of a matrix, and the file features, which are the labels for the rows. And let's attach them, because in R, any matrix or vector can have labels attached to their dimensions. So what I now do here is I read this table, the first one with the gene table, and as you can see, if I read it, I get here this table, and it has given me these 3, this table with 3 columns. Again, for those who are new to R, what you need to know here is that you have to check whether the column has column headers or not. Here we don't have column headers, and you have to check what the separator here is between the column and the tab character. So that's why I say separator is tab. And now I get these 3 columns, and R has named them 1, 2, 3 to be helpful. And I decide that I actually only need the second column, the one with the actual G names, this one here. And I say I want to make the row names of CM with that, and this now means that CM now has row names. If I now look at each row is labeled by its name, and I can now address the rows either by name or by column. The other thing that I now can do is I read in this other file the barcodes. And the barcodes was just a list of values, so I use the read lines function, which just reads each line as an element of a big vector of strings, and that I can directly give to the column names. And now we have row names and column names. And if I now again look at the top first things, then we can see here how the rows are labeled, and for the columns it says they would be labeled, but it suppresses them. So, any questions about this loading in the data? Next aspect that we want to do, we want to make a histogram of the total cell counts. To do this, we first have to think about how do we get the total cell count, and that's of course each cell is a column in this matrix. So if we take the column sums of this count matrix, then it adds them all up, and for each column with its corresponding label, we now get the sum of a respective column. And what I can now of course do is I can draw a histogram. And in R, I simply use the function hist to draw this histogram. And I get this thing here. These histograms, you can give this histogram a second parameter which tells you into how many bins it should divide up the value range. And here by default it's 10. We can tell 30 bins are better. And then we can see much nicer where the mode of this thing lives. Actually, we still see that this bar here contains 4,000 values, so we might get an even larger break and get this nice thing, which now already shows us one non-trivial thing. It seems to low up here a bit to the left. Let's make 300 and we can maybe see this better. Could be true, could also be just noise. The other thing is this distribution has a strong right skew. It has a much fatter right tail than left tail, which is natural for distributions which are limited to a positive support, because of course the column sum cannot be lower than 0 because we are summing sequencing rates. But when we have this, it's often interesting to have a look at its logarithm, and that's what I do now. And we see here the logarithmic column sums. And they look a good deal more symmetric, and they have here a bit a weird left tail which seems to stop somewhere arbitrarily. Anybody notice that? That this thing looks somehow as something has been cut away? The reason being, we used the filtered count matrix. I just gave you the filtered one because the raw column sum matrix is really big. And what filtering means is this 10x machine, it has these little droplets with these beads, with the barcodes, and ideally each bead would sit in a droplet together with one cell. But with a Poisson process, how many cells get into each droplet? So if you feed too many cells, then you might often happen that one droplet contains several cells, and when the transcripts of these cells get mixed, and we would suddenly think that there is a strange cell which combines the features of 2 or 3 different cell types. So to avoid this so-called duplicase, we try to dilute the cells very well, so that it happens rarely that more than one cell ends up in a droplet. But we buy this by saying that most of the droplets will be empty. Because if we look at the Poisson distribution, if I were to make a Poisson distribution where on average I have one droplet per cell, then what I then get is plot 0 to 10, comma, This is the typical Poisson distribution with mean 1, which gives you on average 1 droplet per cell, 1 cell per droplet. And when 40% of the droplets will have 1 cell, 40% will have 0 cells, but quite an appreciable number will have 2, 3, 4 cells. So we should reduce the number, dilute them much more, so that it hardly ever happens that we have 2 cells in 1 droplet, but we count this by having 90% empty droplets, which means when we afterwards sequence our barcodes, most of the barcodes will have no reads associated with them, and only 10 or 5% of the barcodes will actually have an actual read, because only those barcodes were actually in a droplet that contains a cell. The others are not exactly 0, but they still pick up 5 or 10 reads simply because we have such a large number of ambient RNA just lying around. And this means that if we make this histogram of the number of cells per droplet, it looks like this. These are all the droplets which contained a cell, and these are all the droplets which didn't contain a cell, but still got maybe 10 or 20 reads because some ambient RNA which was floating around in the solution got into the droplet. And now we have to cut somewhere here, and this is meant by filtering. You cut it, and then you only store the matrix where the column sum is above that. But the question is where you cut, and there's 2 things that people do in practice. One is that they simply look at the column sums. The other is that they look at the number of detected genes. So they ask how many genes are seen in a given cell, and by seen they mean at least 1 count. Now, question to you, for you to do also something. How do we do this? How can we calculate for each column in our matrix how many non-zero elements are in there? What's the easiest way to write this in R or in whatever language you like? And for the moment, forgot that it's a sparse matrix. This works the same for an ordinary matrix. Now, a thing in these programming languages is that all these matrix elements, there's all these arithmetic functions, when you apply them on a matrix, typically they are applied element-wise. So if I say matrix times 3, then each element is multiplied by 3. And I can not only do arithmetic operations, but I can also do Boolean valued operations, which a mathematician would call a relation. For example, greater than is, in computer science terminology, greater than is an operator the same as multiplication. It is a function which takes 2 arguments and produces 1 argument. Only that this function maps from greater is simply a function which maps from R cross R into the space of true or false. So if I have something like 3, 2, minus 3, 5, 0, 1, like this vector, and I simply say greater than 0 when it says true, true, false, true, false. And now, what next thing I can show you, what happens if I take the sum of such a matrix? Okay, let's give this matrix quickly a name. So this is A. This is A greater 0. If I say, what is the sum of A? It simply sums up 3 plus 2 minus 3 plus 5 plus 2. If I say sum A greater 0, then A greater 0 is this vector, and it sums up this vector. To sum it up, the question is how does sum work with Booleans, and it's very simply all arithmetic operations when applied to a Boolean vector will interpret a true as a 1 and a false as a 0. So what we get is simply 4, because there's 1 plus 1 plus 0 plus 1 plus 0 plus 1. So this is a crucial point that's very important basically for all programming with data. You can always use Boolean operators to get elements to change data into dichotomized data into true and false, or 1 and 0, in an element by element fashion. And it's the same thing I can also do with my count matrix. And if I do that, it just tells us we have a logical matrix, which now simply has the point now stands for false. And with this logical matrix, I can, the same way how I said here, what's the column sum? Right? And it told me for each column simply the arithmetic sum. I can say Cm greater 0, and then it now simply tells me it simply first converts all non-zero values into ones, into true, which it interprets as 1, and then adds them up, and this eventually tells us how many non-zero elements are in our matrix. And if I now make a histogram of this column sum greater 0, and maybe say I want 100 breaks, and actually not. Somehow I was expecting to see a break exactly as by 500, because that was my guess how they made this filtering criterion, but obviously they didn't. Because in Sura, usually if you give it a big matrix, you can tell it, don't read everything, only read the parts which either have a column sum greater x or a column sum which is greater 0, which is above a threshold. But this was just a side note to see a bit how we do this. In any case, with our histogram here, where was the histogram? As I said, we have a histogram like this without or with logarithm. Secondly, let's do it this here. This is without the logarithm. This here is with the logarithm. And in both cases, we can sort of see that the mode of our distribution is somewhere here around 3.7 or 5,000, but there's cells with up to 20,000 and cells going down here. And yes, I don't know how 10x filtered this thing, because usually it would be a sharp cut here at one minimum value, which they might have chosen before making this thing available. So what was our next task? We wanted to log normalize the data. That means we want to calculate for each for each element in the matrix the normalized value, which means we divide it by the column sum. Now again, with the question, how do we do this in R? Who managed to do this in the homework? Okay. What was the trick? Yes. Or did you use another trick?
I multiplied it with the diagonal.
That's of course also possible. It does technically the same thing. You have to think about whether you multiply it with a diagonal from the right or from the left, because multiplying from the right will scale the columns and from the left will scale the rows, or the other way around, I don't know. But if we want to do this, how have you made the diagonal matrix?
I calculated the column totals and then put like 1 divided by the column total for each respective column on the diagonal.
Yes, but with which command did you assemble a matrix out of that?
Oh, diagonal and then...
Exactly, so you used the diag command. And if I now did this here, diag columns count matrix, then it gives me this matrix. And the catch is, this is now a dense matrix. You see all the zeros are written out. 5,125 here. And here this works, but in practice we might run into the same thing again. Now R will internally allocate a big amount of money of memory to store all these zeros, and we might run out of memory. And whenever you work with big data, you always have to be mindful of, am I wasting memory at the moment? Actually, you don't always have to be wasteful. Sometimes it is a better strategy to just go ahead and do whatever is simplest, and only if that will fill up your memory, only then to switch over and think about it, how you can save memory. But once you come into this, you might get into this issue. And so this is why I mentioned why we might be better off with this transposing idea. The transposing idea, by the way, is necessary because in R, if you have something like, let's say we have the values 1, 2, 20 here, and I put these values into a matrix with 5 columns. Then, as you can see, it fills up the matrix column by column. This is called a row major matrix. Other programming languages fill it row by row, which is then called a column major. This here is called row major because if you have an indexing, when going from one element to the next, the column index makes smaller steps than the row index, or makes faster steps. This is why we call this row major. And in a row major thing, if I now were to multiply this with values like 1, 10, 100, 1000, then you see, because what it now does in order to perform this multiplication, it goes through the matrix in the order in which it was filled and say this times 1, this times 10, this times 100, this times 1000, 1, 10, 100, 1000. It even does something strange if I do it like this, when it always goes 1, 10, 100, 1, 10, 100, and you see it's offset, and in the end it has a value left over, and it just warns me briefly that somehow it didn't quite end up, but it still does something, which is a bit a dangerous thing with R. But in any case, it means that we always get the rows multiplied, and if we want to get the columns multiplied, we better transpose the thing. I transpose it. Now I have the values like this. Now I can get the values multiplied with 10,000 different, and I can transpose back, and I get what I wanted. So we do this now for our count matrix. I transpose the count matrix, divide by the column sums, and transpose back. Now, transpose the divide by the column sums and transpose back. And then we get this matrix. And I call this now maybe the fractions matrix, Fm. Now if you look closely here, the fractions matrix is no longer a DGT matrix, but a DG3 matrix. Let's have a quick look what the difference is. This is our count matrix, and as you remember, it stored its data with 3 vectors I, j, and x. And now let's see what the fractions matrix done, because somewhere on the way R has internally went from a triplet sparse matrix to a column sparse matrix. And the column sparse matrix you see here, it seems to need less data. Instead of 3 long vectors, it now only has 2 long vectors. And the third vector is smaller, and it's no longer called J, but it's called P. How is this possible that this matrix still contains the same amount of information? The reason is here, by the way, for the Germans, the audience, if you ever read a German textbook, sparse matrix is usually called a dünn besetzte Matrix in German numerics textbooks. So, and here we have now an example of a matrix which is sparse. It has only elements here, 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 and here. And in the easiest case, you might have now let's take this matrix here. Here it's easier to see. It has in the first row it has 2 elements. In the second row it has 2, and so on. And I can now either write this in triplet sparse by saying element 1, 1 is 10, element 1, 2 is 20, 2, 2 is 30, and so on. But I can also store it as following. I simply write all non-zero element values in row order in it. So 10, 20, 30, 40, 50, 60, 70. And for each of them, I write in which column I found this one. This in 1, 1, 3, 2, 3, 4, 5. And then the last thing is I wrote down where I started a new row. The first element is in new row. The element number 0 is in new row. The element number 2 starts in new row. The element number 4 starts in new row. The element number 7 starts in new row. And then the 8th new row, element number 8, doesn't exist anymore, so this indicates that now the rows are finished. So basically for each of the 4 rows here, it tells us where the values for this row start, and then in the end, just by convention, we add a marker which tells us where the end of all of them is. Yeah?
What do we do if we skip a row?
If you skip a row, when you repeat the index here, you would, for example, if we didn't have a row, 3, then here this element would be 7. Because, no, not 7, but basically you duplicate. If you see twice the same value next to each other, then you know, because this here you can also read as the entries from 0 to 2, not including the end, are the first row. Rows from 2 to 4, not including the end, are the second row. In verse 4 to 4, and you read this as the elements entries from 4 to 4, not including the end, are the third row. When you read this, the third row is empty, because when you exclude the end, then you don't have it anymore. This is a typical case of fence posting. And you might know, if you've ever worked with Python, you might know that Python has this habit that ranges are always written start included and end excluded. And this is precisely the reason why you do that, because all these things get a nice natural logic to it if you fit to the convention that all the intervals you ever specify do not include their end, because then you have a natural way of specifying an empty interval. R switched while we transposed without being asked to from the triple sparse to the column sparse format. The reason for this is that R prefers the column sparse format because it is the preferred format for matrix vector multiplications. And the reason for this, if you imagine you want to multiply a vector from the right to this matrix, when you get a result vector, and you want to calculate, say, the third component of a result vector, then you can immediately say, in order to calculate, I shouldn't say the third, other terminology thing. In computer science or in software engineering, we have to be careful with ordinals. We shouldn't say the third element, because in English, the third element implies that we start numbering with 1. So this here would be the third element, but we number by 0, so we call it the element number 2. And it's better if you use a zero-based scheme to avoid ordinals for this region. So let's say I want to calculate the number 2 element of my result vector, when I immediately see, okay, the information I need that is in the indices of 4 to 7. So 0, 1, 2, 3, 4, 5, 6, not including 7. So I have to multiply these 3 values with the elements 2, 3, and 4 of the incoming vector and add them up in order to get the element number 4 of the outgoing vector. And this is why this column sparse is the data format optimized for performing matrix, and matrix vector multiplications. Obviously, all this is not so important to do the biology side, but it's very important if you try to really do high performance computing with scientific data, that you know about such subtleties, because that makes the difference between code running a month and code running an hour. So But here always happened automatic, and now we have our F matrix. But still, whenever I do this, I feel nervous whether I have done all these transposing and transposing back correctly. So the first thing I now do is that I quickly check the column sums of fraction matrix and check whether they are all 1. And luckily they are, so I've done it correctly. Now Fm is normalized and its entries add up to 1. And now, what I can now do, I can take this fraction matrix and take the logarithm. And if I now do this, something annoying will happen. Because all the zeros, so far the zeros have stayed zeros. If I look into this fraction matrix, it still only lists the non-zero elements. And that's good because when we divided the zeros by the column sum, they stayed 0, and R was clever enough to notice this, or maybe not R, but this underlying C library for sparse matrix, noticed this and knew that zeros stay 0, so it can skip over all the zeros and only look at the non-zero elements here in the ingoing matrix. But if I now take this one here and calculate the logarithm, all these zeros will turn in minus infinity. And because minus infinity is not 0 and all non-zero values have to be stored explicitly, our matrix will blow up to gigantic proportions because all the zeros now have to be stored explicitly. So if I now press enter, my computer will crash again. So I don't do that. Instead, I call the function log1p, because the log1p function is again a function which preserves zeros. As I told you in the here in the homework, log1p is the function defined as log of x plus 1. And this function has its own explicit implementation for 2 reasons. First, it's a zero-preserving function, and whenever we work with sparse matrices, we prefer using zero-preserving functions, which map 0 to 0. There's another reason why in all arithmetics libraries for computers you have 2 separate functions, one for the logarithm and one for the logarithm of x plus 1. Anybody has an idea why? Hint, it's a calculus question. What is the Taylor series of a logarithm around 0? Anybody remembers your Taylor series? Everybody notices that this was a trick question?
The only logarithm or the best logarithm we can make is around 1 plus x?
Exactly. Yes, because we cannot write down a series expansion around 0 as we would do for sine or cosine, because the logarithm is undefined at 0 after all. So we have to do it anyway, and the easiest way to do it is the series expansion around log 1 plus x, because this series expansion is, as you might remember, simply, and now let me better look it up, log 1 plus x. I think it's simply the harmonic series expansion. I don't need quora here. X squared half plus x thirds 3 plus and so on. Look, there's even a YouTube video about it. So this is a terribly easy expansion. It's also a terribly well-behaved one, because at that point the logarithm has slope 1 and is sort of most well-behaved. So if you want to calculate the logarithm, you lose a lot of precision if you calculate it exactly, even though you actually want to do this. So you always go through this thing. If the computer, when it actually calculates, it shifts your argument so that it can use this Maclaurin series. And this is the reason why it's implemented. It's the actual reason why there's a special function, but it comes in very handy now to work with our sparse data, because it allows us to quickly calculate this here without my memory blowing up. The catch of this is, of course, that this normalization doesn't quite do what I want. I can demonstrate this by taking a random cell, column sums. Let me look a bit at the column sums. Let's take Let's take cell number 123. And now I take here, this is everything that this cell here produces. And I want to compare this with log 1p of this. 2, 3. And no, it happened again. Let's see if my... Nope, it's crashed. So, but anybody already knows what will happen if I now do this? Who can make a guess how the graph will look like? While I restart the computer and go back to where we just stopped, you can make a guess about what the result of my little plot would be. Actually, I wanted to use FM 1, 2, 3. So I take my random thing here, comma log 1p, this thing, and plot it. And what we get is this here. The logarithm hasn't done anything. Why is that? How can this be that my logarithm, this doesn't look like a logarithm. Logarithms are curved, convex curves. Who knows what happened? Nobody? We have small values here because we divided the matrix column by its sum. So all the values here are smaller than 1, and they are a good deal smaller than 1 because we have a few thousand genes there, and we need to partition the 1 over all of them. They were all around 0.1, and now remember log 1 plus x is, as we've just learned, what did we just learn? 1 minus x plus x square half minus x cubed 3 factorial plus and so on. Was it factorial? No, it was without factorial, right? So these things usually blow up here because that was the whole point of a logarithm. It does a lot of things. But now we add a small value x to plus. So this x here, these are the x's because we calculated log 1 plus. And we have here 0.005 squared, 0.005 cubed. So of course this won't change much, because if we look at the curve of the logarithm, here at 0 it goes through slope 1. I have to find a nice logarithmic curve. We are sitting here and we have shifted our 0 here because we added 1. And now we add a little point here. Oh, 1, so let's say here is 1, here is 2, here is 3. And now 1 plus x, these are all values here. And that's the very point where the logarithm is most linear. We stay in this little bubble where the logarithm is really well approximated by this line. Actually, physicists might remember this kind of approximation that we always say. Log 1 plus x is about x for x more than 1. The same way how you might say sine of x is above x for x smaller, very small compared to 1. Who has used these ones? Exactly. So we fell exactly for this. The approximation that we always use to make our calculations easier now cause our logarithm to have no effect. That's annoying because, but now we do a simple trick to solve this. Our actual issue, so that we can still use the logarithm as it is intended, is, well, if this is too small, let's blow it up. Let's take, instead of 1 plus x, let's take 1 plus 10,000 times x. And now it looks like a normal logarithm, because what happened now? We have done log 1 plus 10 to the 4 times x, and this is of course the same as log x, the same as log x fourth as soon as x is very big, is sufficiently larger than 10 to the minus 4, this stuff here will be sufficiently larger than 1, and we can neglect the 1. So essentially what this thing here really does, it is something which for large x acts like a logarithm, but for x around 1 or so, or let's say x much smaller than 10 to the minus 4, when we get again into this realm, and it acts like an x. So it's a smooth interpolation between a linear transformation or an identity transformation for, sorry, not x, but 10 to the 4x. So for a very small x, it's simply a linear scaling, and for very large x, it is a logarithmic scaling. It smoothly interpolates between a linear and a log transformation. And the point where it switches from one regime to the other is around 10 to the minus 4. Why have I chosen 10 to the minus 4, 1/10,000th? Well, if you go back to our histograms, Rolsam's hist log 10, Rolsam's count matrix. You see all our values, most of the Rolsam values are around 3,000 and maybe 20,000. So 10,000 is smack in the middle of this thing. So if a gene has 0 counts, well, it gets mapped to 0, that's what we wanted. If it has 1 count, then we are sort of in this linear regime, but once we have more than 5, 6, 7 counts, we get into the logarithm regime. And that's good because that makes a lot of sense. We can have a look here at this one again and see what does this thing do if instead of 10 to the minus 4, we take 10 to the minus 2. Now you see we don't get a proper logarithmic scaling. But we wanted a logarithmic scaling because in the end, gene expression is best looked at on a logarithmic scale. That's something we haven't really seen yet in the lecture, but we will see that whenever you compare expression of one gene over many cells, you will notice that it has a log normal distribution. So in biology, everything looks normal once you take the log, which by the way has an interesting reason, namely, you all know the central limit theorem. Or who knows, remembers what exactly does the central limit theorem say? Or not what exactly, but what approximately. Roughly, the central limit theorem says, if you add up a large number of independent random variables, the sum will be normally distributed, right? That's the whole point of a normal distribution. The crucial point is adding up. If you add stuff up, when the sum will get a normal distribution. But in biology, everything is controlled by biochemical reactions. And in biochemical reactions, the speed of a reaction and the amount of product that is produced depends not on the sum of the concentration, but on the product of the concentrations. You might remember this as mass action laws of the Germans as Massenwirkungsgesetz from high school. You always, and because all the biochemical products in your cells, their concentration depend on a reaction and therefore depend on the product of the precursors, and the concentration of these products of these precursors depends on the products of their precursors, and so on and so on. Everything in biology is actually not the sum, but the product of very many independent factors. And therefore, the central limit theorem tells us that not the concentrations itself, but their logarithms will be normally distributed. And this is in core the reason why in biology we always find out that actually we have to look at the logarithms of the values for things to make sense. And that's the reason why we actually want to have a log transformation of our data. But the log transformation causes all these problems with the zeros, so we try to interpolate. And here we have not enough log. This is still practically linear, but we wanted a log. So now I take here a too high thing. And now I have a nice logarithm, but I get a giant difference between 0 and 1. You can see it now, but this point here is probably these are genes which have exactly 1 count, and these are genes which have exactly 0 counts. And actually the difference between a gene that has 1 count and a gene that has 0 counts is pretty unimportant. Because after all, if a gene has only a single read count, then it's quite likely that if we were to look at the same gene in the same cell again, we would probably miss this thing because it's probably completely random whether we see this one single molecule or don't see it. So the difference between 0 and 1 shouldn't be too big. It definitely shouldn't be larger than the difference between 1 and 2. I cannot make it smaller because you can easily prove that this difference will always be larger than that difference. But I can at least get them on the same footing. And on the same footing, I get it if I use this here. Ideally, I would use exactly the value which I have, because 1 0 read count is mapped here to 0.0002 or something, to whatever the column sum for this cell was. And so I should use the column sum for this cell here as the scaling value. Then this difference between 0 and 1 will be the same as those between 1 and 2. But that is not good, because then I would use a different scaling factor for each cell, and I can't compare the cells. And the whole point of our normalization is to make them comparable. So I have to find a compromise. And for the compromise, I maybe best would take the mode here of our histogram. And in practice, the mode is always a little bit different, and so that you can also compare between different experiments. The field basically has settled. It only has to be roughly mode. It doesn't have to be exactly the mode. So let's just take a nice round number which lies in the right area, and that's why everybody uses 10,000. So as you can see, behind this harmless little formula here, there is a little bit of not too deep but also not completely obvious math, which in the end is something about the series expansion of a logarithm. So, questions about this now?
Could you show the code where you are plotting? Where it doesn't work in my case.
What does it do? What have you written?
I'm not work with R, so.
Let's have a look. If you want to debug something like this, what you typically might do is no. You don't mark the whole thing. So what you're doing at the moment is, let me copy this quickly into this is maybe a good also general thing. if you have a code piece like this and somehow it doesn't work. And when what I do is when I look at the individual parts, I might say, okay, this plot command takes an x and a y coordinate. Does the x coordinate work? I mark this, press Ctrl Enter, and I see, yes, the x coordinate produces a nice vector. Does the y coordinate work? In that case, it does. If the case it doesn't, then I can ask, is the argument inside that thing? So if you want to debug in R, it's a good idea to go outside in or inside out, mark parts of your expression and check which term actually produces the error. So our next job is to find highly variable gene. So for each gene we want to calculate its, sorry about this, of course I meant it's normalized but not logarithmized expression, because here I told you Pij is, I hope this didn't confuse anybody. Yeah, because that's the point. If you write formally and words and they don't agree, then you don't, then probably the formula is correct and words not, but you can't be sure of it. Yes, this is what I meant. We normalize, but we don't logarithmize, because that's what I've written here. So I take my fm, which you remember, maybe just to keep what we've done here. So this here is our fm, and we take now this normalized things, because now with genes, the cells are comparable, and now we can ask for each gene what's the average expression. For example, by saying, what is the row mean of this? And now I can ask questions like, which are the most highly expressed genes in this data? To do this, I simply sort my row means. Now I got all the lowly ones, so I look at the end of this thing and find now the most highly expressed genes. The most highly expressed gene seems to be MALAT1, which takes about 5% of all the reads in a typical cell. And then this one, which takes about 1.5%. Okay, so this way I get my row means. And I also want my row vars of this fractional matrix. Row means, row variances. Now we get an annoying issue. Who got this error message? At least one. All the others didn't get it. For the others it worked automatically. The reason why it doesn't work is you first have to load this library sparse matrix stats. And without that it doesn't work. So we need these two functions, and without them we don't get far. Yes. So make yours. There is a row vars command for normal matrices in R always, but the one for sparse matrices have been pushed into this strange package here. And with that I can now plot the means and the variables, and now I can plot one against the other. Plot row means on the x-axis, row vars on the y-axis. Who got that far and got this plot? Okay.
It's got a different plot.
What is different?
It looks
Now, noticeable, of course, this is not the plot I thought I had. We wanted to get to this plot eventually, right? But what's missing? Yeah.
For the other one, you want to divide the variances by the means?
Yes, this is one thing. And we can try this. I divide the variance by the means. What else is missing? Well, actually, if you didn't know what plot to get, but you get this plot, what's the first thing you notice?
It's not logarithmic anymore?
Yes. Or the other way around, if I see a plot like this and I look where are the x values, you notice well, there is a lot of white space on this plot. All the data is in the bottom left corner. I could now have the idea of zooming in this area, but actually I already know that probably I should logarithmize because we need it all the time. So we simply tell R to make the x and y axes logarithmic. And now you get them like this. Or if you don't divide by the mean, it looks like this. You might know a subtle difference. What I just have done here, I can of course accomplish two ways. One way is to tell it make the x and y axis logarithmic. Another way is to, let's say you didn't know that, you can make this log equals xy argument. You might have had the idea to instead do this here. What's the difference? It's still the same plot. The difference is only how you scale the axes, whether you label the axis with the logarithmized or with the unlogarithmized values. If you take the log yourself, then R will of course put the logarithmized values because that's what you give it. If you tell R to take the logarithm, then it will keep the labels unlogarithmized so that one can see that the axis is logarithmic. Yeah?
If I don't include this sparse matrix package and I put as matrix before the matrix, is that a mistake?
This works, but again, it will convert your sparse matrix into an ordinary matrix, and depending on how much RAM it has, it may or may not blow up your computer and crash R. This is exactly what I meant before. Sparse matrices save a lot of time, a lot of space and compute time, but they have a problem that, of course, all the computational routines have to work differently because the data isn't anymore where it's expected to be. So you need for all the little things like means, variances, multiplications, and whatever it is, you need new code. Now, of course, in practice, this problem is solved. Some people in the 1970s have spent a lot of PhD theses writing all this code, and now everybody uses that. You only have to make sure that R knows that it should use this code. So here now I have my plot. Let's use this one with the logarithmized x-axis. The next thing I do at this point is I look at this and say, there's so many points plotted on top of each other. I don't even know whether, what is there? Is there many points or only a few? So how would we solve this? The first thing is, of course, to say, make the points a bit smaller. We are here big circles. Just make them only a tenth of the normal size. That's what the sex argument does. And the second thing that you can do is that you say, and this is maybe a bit more subtle, that you say, make them transparent with only 20% saturation. So color is black as before, but with and now the points are semi-transparent, and it's easier to see something, to see how many points are there on top of each other. I usually then like to try around. Maybe I make them only 5% covering, and now all these individual points are really faint. I can only see those which are really fat, or I make them 20% when I can still see all points. But that way, this is a simple, cheap trick to make sure how much overplotting you have to play with this alpha channel. The better way, if you don't want the cheap, but the proper trick is there's also functions which then count how many points are plotted on top of each other and use a different color accordingly. So, and I do the same thing here for my other plot, where I divide by the where I divide by the row means, and now I get this plot here. So, now I put in this purple line, which is here, it goes through the middle. How do I calculate the position of this purple line? The expected Poisson noise. Who has managed to get the purple line? You know, that one here. How did I know where to put it? That was the thing we did last time in the lecture. Let's have a look at our lecture again. Where we asked this question here with our container with all the balls, and that's the situation we have here. Each cell is a container, each transcript is a ball, and of course we have here a Poisson distribution which tells us how many balls, red balls, so balls from transcripts from the gene we are currently making our dot for in the plot, how many of all the transcripts we get, so the column sum is all the balls that we draw, and the red balls are the balls which are the one matrix element for which we are currently making our point. And we ask how are they distributed. In the lecture we have found that they are distributed according to this formula. Q naught over n tilde plus V. And what we did here, you remember we looked at these PI, which are exactly our normalized but not logarithmized counts, our fraction matrix Fm. That's what I call P in the formula. And Pi is the P now for cell I. And when we calculate the row vars of this fraction matrix, we calculate this W. This W is one of the rows of Fm. So each of these row vars here, this thing here, this is such a W. And the same as in the lecture, we calculate, we take this variance going over all the elements, and each of them is a count which has been normalized with a different column sum. That is what we had here. Ki are the counts and ni are the column sums, and I is the column index. And we've seen that the expectation for this thing is Q naught over n tilde, where n tilde is the harmonic mean of the column sums, plus the actual real variance from one cell to the next. The minimum that the real variance can have is 0. So this here is the minimum, this here is the smallest possible expectation for our gene. The gene's average expression divided by n tilde. So, in this gene here, in this plot which I have up here, that one, I can now say for each row mean here, the row means that are the Q naught, and the row vars that are the W. And now we say the expectation of a W is Q naught over n tilde plus a positive value, where the minimum is 0. So we now have to draw a line which goes here. Let's make here a line from -9 to -2. Out 100. So you see, these are lots of values which simply span the x-axis. I call this x grid because these are all values which span the x-axis. And if I divide all these potential mean values by n tilde, then I get the expectation for W. And what is n tilde? N tilde is, as we've just seen here, the harmonic mean of the col sums. Ni is the col sums, so we have the col sums of our count matrix. We take 1 over this, and then we take the mean of that. This here is this formula here written in accounts. And now I can here say points xg, xg divided, Kroll gleich purple. And let's make small points. And what do I get? I get a purple line which is at the wrong place, because here is my FM, here is my plots, and why are they now up there?
Shouldn't it be not the count matrix, but the fractional?
No, it's the count matrix. That's exactly the catch here. We divide by ni, and ni are the number of balls we draw, so the number of counts we get out of this. Fraction matrix, count matrix. Ah, now I see it here. This is it. It's 1 over the mean. It's the harmonic mean. I've forgotten this 1 over here. You see, it's a harmonic mean, so it's 1 over the mean of 1 over the values. And now the purple line goes right through where it should go. And now that we know that, we also know that we should make this plot here where we divide by Q0, because if I divide by the row was, so if I divide each y value by what was the x value, then of course here I also divide by the x value, and then I'm just left with this one here. Which is now just a horizontal line which goes straight here. So I use my AB line and say make a horizontal line here. So to sum up, we now put basically this lengthy calculation in code and said if we have our count matrix, we can normalize them by dividing each column by the total number of counts for this cell, and then we can plot means against variances, and our expectation for the variance is always simply the mean divided by n tilde, and this mean divided by n tilde is exactly the value which goes here through the bottom. And as it's mean divided by n tilde, we can divide the whole y-axis by the mean, so that we get the other plot, that one. Does this make sense? It's one of these things one has to think through a couple of times. And if you want to go away from the biology for the n tilde, you can also go back to the lecture where we use this completely nondescript containers with balls to show that this has nothing with a specific thing, but it's a very general mathematical thing. That we get whenever we take we check concentrations from many different items. So, now that we have this, we can now say that all the genes here at the bottom, all of them, they are obviously uninteresting genes, or genes which vary not much because their V here, this V here is small, so that they lie on that line. And if a V is small, which means that the actual concentration of a transcript in the cells is the same in all cells. And if it's the same in all cells, it can't tell us anything interesting about the differences from one cell to the next. Those genes up here on top, they are different. They might tell an interesting story. So we now try to find the names of the genes here on top. So how would I do that? Anybody got an idea? How do I get the names of the genes which are at the upper end of a plot? Yep.
Something like print F or something of the
genes list, you just list all the variances in a.
Okay, I take all my variances, rho vars fm. Here we are. And what next?
You sort them.
I sort them.
Then you pick the top. I think you told us to pick the top 1,000.
Okay. So I take the first 1,000 in the list. They are all 0. Why are we all 0? Yeah,
Because it's sorting from lowest to highest. So
exactly.
we have to reverse the order.
Yep. Or we have to or take the last one. So we should reverse the order. We can check in the help for the sort command and see, there is a decreasing thing, and we have to say decreasing is true. So, and now we get these genes. And we have their names here. Now this is a named vector, so it actually has all the values as elements and has these names. What you can do is that you say, what's the names of these? And then you get the names. But before we take this, let's have a quick doubly check. Will these names list really be the names of the genes which are up there? Who sees the mistake that we made? Check the code that I have marked here and check the plot again. We want the names of the genes which are in the upper half of the plot. Yeah?
Is the row variance divided by row means?
Exactly. At the moment we have only the row variance, so we would take the top things from this here. But that's not what we want. We want the top of these. So of course we have to sort this plot's y-axis. And the y-axis of this plot is rho vars over rho means of this. And now we get our highly variable genes. Let's store them. I call it HVG for highly variable genes. So. Who got these? I gave you the first couple of them. Is this the same that we now have? Let's check. HVG, and where does it start? Here. IGKC. So who got to this list? Okay, we are getting there. I mean, if you don't worry. I mean, this is all new, especially if you're not used to R, but this is the kind of exercises which really make you a proficient and competent data scientist. So maybe just try again in the next days. So now we have found them. The first thing which I now want to do is to look at them, because we said these things might tell us about differences. So let's pick one of them. Let's maybe pick here the gene IGKC, which happens to stand for immunoglobulin kappa chain. And let's have a look at the immunoglobulin kappa chain. And I want to get a histogram of its normalized values. How do we do that? Make histogram of log normalized expression of IGKC. What do we do? Which of our various data objects here do we have to look into?
The fractions matrix?
The fractions matrix, yes. Okay, here's a fraction matrix. How do I find the gene I want in it? What do we have to subset? Do we have to find a row or a column or a square, or what do we have to find? Yeah,
Say we look for the specific row named IGKC.
exactly. The nice thing in R is you can always just use square brackets, the comma, and then to the left of the comma you say the row, and to the right the column. And a nice thing is we can either say, for example, give me the third row, or give me the row labeled with IGKC, like this. And now this here is what we have. So, FM IGKC. And now I want to make a histogram of that, so I just type hist. Figure margins too large. If you ever get this error, it just means that you have to make the plot window bigger. So here is my histogram. Doesn't look very nice. What should I improve? Say again?
The breaks.
Okay, let's make 100 breaks. Looks a bit better, but what should we do? Yeah,
Is there a way to like cancel out all the zero elements?
we could do that, but then maybe we get this. Let's try this. What I could do is I simply say ylim is C0, 100. Now we can see the rest, but still somehow it doesn't really help. What's the other thing we've forgotten about? Yeah,
The logarithms.
the logarithm. We've talked all this time about how to best do the logarithm. Now we should do it. Now if I do it like this, so I use my normalization formula that we had. Log 1p of this thing times 1e4. That's what we did before. Entfernen. So, now let's make 100 breaks. And now let's add what we did before with this ylim is C0, 100. Maybe 0, 300. Now we have got a quite reasonable histogram, right? Now we can read something. There's a lot of zeros, but among the non-zeros we see two populations. Probably even three. There's here this population which is very slowly expressed, this is a bit, and this is the third one. And this is how we now find biology. Now we've already learned that the gene immunoglobulin kappa constant is at least bimodal, if not trimodal. So the cells are split into two parts, those cells which express this strongly and those cells which express it weakly. To get a better feeling of what this is, let me switch this quickly to this. Now this time this is exactly the same. I don't have to care anymore about the problem of preserving zeros. So instead of log 1p I added +1 here. And the reason why I did is so that I can switch from natural logarithm to decadic logarithm. Because now I can easily read. We have done here 10,000. So 0 means 1/10,000th of all the reads fall onto the cells. This is 1,000th fall onto it. This is 100 fall onto it. So all these cells here have a full 1% of its transcripts falling onto these genes, while the others have less than a per mil. So now we've learned that there's two populations of cells. One population, a smaller one, which has about a % expression of this gene, and all the others have less than a per mil. And we want to have a bit more systematic way to find these things. So we keep this in mind, but go on a bit more. And now we do our PCA. For the PCA, we need to, it's already 15:15. Well, let's quickly go through the rest of the code. We now have our Fm matrix, and what I want to do is I want to switch it to logarithmite it. So I take the whole thing, log 1p of Fm times 1e4, and let's call this ym. No, sorry, log 1p. Now I have this one, and because I really hate this stochastic logarithm where I never quite know how to read that, I now divide it by log 10, so then it's basically a logarithm to basis 10. So what I've done here manually by switching from the natural to the stochastic logarithm, I have now done here manually by dividing by log 10, which then rescales the logarithm to the other basis. Now I have this ym, and what I now want to do, as you've seen here, I want to center and scale it. So for each gene, I take the mean of this one, row means, and I subtract it from the row. And this time I don't need to transpose because it's already on the right side. Because it is already the rows, it's not the columns. And then I divide by the row standard deviations. Row vars ym. Like this. And I call this now yc, the centered variable. And again, something is happening here. This time I seem to have made a mistake because now I got my big matrix again with 3.4 gigabyte, but this time it has fitted in. I know why, because when I subtracted the row means, all these matrices became dense again. What I can now do, I can now calculate pre-comp. I can now use this IRBA package, which can calculate a PCA like this, YC. And if I did this, it would take, and the IRBA package, I can tell it whether it should center and scale the matrix, and I don't have to do this, don't want it to do this because I've just done it myself. But now while I'm saying this, I realize that this is bad because YC is now a dense matrix, and it has to work through this dense matrix, and that will take ages. So what I better do is I give IRBA directly my unscaled matrix and tell it, please do it for me, because then it can work with a sparse matrix. So I do this. I forgot to tell it that I want the 30 top principal components. And this now takes a moment. Stop. And as you've seen here, I told it that I want to transpose the matrix because a PCA, that's what we seen yesterday in the lecture, you also remember, I started explaining to you all the data are in the rows because that's statistical standard. And then we switched it over because PCA wants it the other way around. So now PCA works a bit, and usually it goes much faster, so something is weird today. Let's give it a moment, and if it doesn't work when we are out of luck. But actually, what I simply can do here is, because I showed it to you here, I stop it here. Wait a second. Yes, terminate R for the nth time, and when I quickly show you now, because you probably want to leave because I'm already 10 minutes over time, I quickly show you the code in my already prepared stuff, and we can finish there. So again, we load the count matrix as we've done last time. We load the G names and the barcodes, when we apply the gene names and barcodes to the count matrix. Now we have our column sums. We divide by the column sums here. When the next thing is we calculate the log transformed values with the log 1p. And finally, now we calculate the highly variable genes. Here they are. And now I center the matrix. I remember what I've done wrong. This time I didn't, because why did we take the highly variable genes? Because I didn't want to wait IRLBA to do the PCA on this giant matrix with 20,000 rows. I wanted to use only the top 1,000 rows. So here I've taken my matrix and I subsetted it to only these 20,000, to these 2,000 things, and this is why now by my new matrix YC is only has only 1,000 rows instead of 20,000 rows. And now the IRLBA package will be quite fast, no matter whether the thing is sparse or not. I give it this thing, let it calculate its PCA. And now we can plot the PCA. Here, for example, the first two components. And we can see that now our data has split up into two populations. And maybe one of these is the population which expresses the gene that we've just seen, the other is the one which doesn't. And finally, I gave here this matrix with the PCA coordinates. You see, PCA gives you back here the list of standard deviations. That's what we looked at yesterday. These are the eigenvalues. Here the rotation matrix with the eigenvectors and PCA. And these are the eigenvalues. These are the eigenvectors. It's called a rotation matrix because essentially it takes our subspace and rotates it into the basis that we have chosen to use in the basis. And X is the result of a rotation. So this is now the result of a projection. For each of our 11,000 cells, we have now a projection into this 30-dimensional space. And we take the first two of these 30 dimensions and plot them. Here, and then we get this. And now I give these 30-dimensional projection to the UMAP algorithm, which is this algorithm which now tries to find places which are small and plot them. And now I get this plot, which tries to summarize the 30 dimensions to 2 dimensions. And I can use the sleepwalk tool that I've already showed to you once. Okay, it says sleepwalk. No longer here because I've installed it. In a second. So. And we can see here how this worked. UMAP has properly put all the cells which were close to the cell under my mouse curve in this 30-dimensional projection space on this side. And here, this bulk is everything here, and this is another blob, and so it sort of worked properly to split this up into different things. The next thing which we now could do is check in this plot which of the cells have this this gene that we were just interested in. But let's stop here. So far, we have now seen how we organized our data, how we got from this count matrix to this thing, where we now see subpopulations and can say, this is probably one cell type, this is another cell type, and this other cell type seems to fall into 1, 2, 3, maybe 4 different subpopulations. And now we can ask, which are the genes actually that separate, which are different in these cells compared to those cells? And when asked our biologist colleagues, we have found that these two cell populations differ in those genes. What might this be? Questions about that part? then I guess those who had difficulties, it might be a good idea now that you've seen how it's done. I will now simply unblind the code that I've hidden before in the homework and put it in so that you can see how it works. And then you can compare it again. But what I would encourage you, I put this separately so that you can first use the blinded part. Try again. If you don't manage, you can cheat and look into the unblinded version and check it out. But yeah, here the good thing here is because it was basically about handling big matrices, transposing them, normalizing them, getting the column and row sum, scaling them, all these things. And in the end, we get our PCA. The other thing I wanted to do is make a little exercise where I ask you to play with the big 5 data that we talked about yesterday. The exercise will essentially be do the test, then project yourself into the 5-dimensional, take your own test answers, project yourself into your 5-dimensional space, then predict from your own 5-dimensional values how you should have answered to the question and compare with what you actually did. Obviously, better keep your actual answers for yourself in case they are embarrassing. We want to obey data protection standards here. So, then I see you all next week.